Hello and welcome to this Australian Biocommons webinar. My name is Melissa Burke. I'm the Australian Biocommons Training Manager and I'll be your host for today. This webinar is part of a series developed by the Community for Structural Biology Computing in Australia, and it focuses on leveraging deep learning to design custom protein binding proteins. This webinar series has been designed to inspire and help you recognise the potential applications of these new approaches by showcasing some of the ways that Australian researchers are putting them into practice. Before we begin, I would like to take a moment to pause and acknowledge the traditional owners and their custodianship of the lands on which we meet today. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country, and we recognise their valuable contributions to Australian and global society. Today, I am joining you from the lands of the Turrbal and Yagara people in Brisbane. So we are thrilled to welcome Dr. Richard Birkinshaw to our webinar today. Richard is a senior research scientist in structural biology at WEHI, where he runs the WEHI SPR instruments and is establishing WEHI's AI protein design platforms. Richard's interests revolve around furthering our understanding of disease and leveraging this to develop novel targeted therapies for cancer, developmental diseases, and viral infections. Today, Richard is going to be talking to us about using in silico design methods to create de novo proteins that selectively modulate apoptosis. Welcome to the webinar, Richard. I'll now hand the screen share over to you to start the presentation.
Thank you, Melissa. Okay, great. Thank you very much for inviting me to talk today. I would also like to acknowledge the traditional owners from where I'm speaking today in Melbourne, Naarm, that is the Wurundjeri people of the Kulin Nation, and I would like to pay my respects to their elders past, present and emerging, and recognise that this land is unceded. So today I'm going to be talking about some work we've been doing. This is going to be broken down into two parts. First of all, selectively targeting the apoptosis protein back with de novo proteins. This is more of a sort of traditional approach to designing proteins using machine learning. And then in the second part of the talk, I'll be talking more towards these deep learning approaches and how they're being used to design proteins now, and how one of the test cases that we have done here, or I have done here at WEHI. So a bit of background on the diseases we're interested in. Apoptosis is a fundamental process that we have in life. It is the targeted deletion of or death of cells, and this is probably most evident in the removal of webbing during interdigitation, as shown here on the left. Here, where red cells are cells that are undergoing apoptosis and are slowly dying. And it's an essential process for multiple things, including host defense against pathogen, also in cancer prevention. And this is why we're particularly interested in it, because we now have approved therapies that target apoptosis, specifically the BCL2 family of proteins, these pro-apoptotic BH3 mimetics. So a little bit of background on the BCL2 family of proteins that we're particularly interested in, and BAC is a member of. These are broadly classified into three families by function and structure. You have the BH3 only proteins. These are largely unstructured with a single alpha helix. They promote apoptosis, and then keeping these in check, we have the pro-survival BCL2-like proteins. These prevent apoptosis progressing. They have a full globular fold consisting of 8 alpha helices and a transmembrane single pass alpha helix. And then the third class, which BAC is a member of, is the pro-apoptotic effector proteins. And these have a combination of the two. They promote apoptosis by unfolding and causing membranes to break. But they start in an inactive form that has the BCL2-like fold consisting 8 alpha helices. We have multiple members of these different families. Most relevant to today's talk in the pro-apoptotic group is BID. So that anytime I mention BID during this is normally a protein that will promote apoptosis. We then have the pro-survivals relevant to this talk, particularly as MCL1, and I've already mentioned BAC as a pro-apoptotic effector. And the way they work is there's an interplay between these different classes, such that when you have a steady state, there's low expression of BH3 only proteins and a relatively high level of the pro-survival BCL2-like family proteins. These two interact. And so when you get an apoptosis cue, you get overexpression of these BH3 only proteins that saturates this pool of pro-survival proteins. And then these BH3 only proteins can go on to interact with the pro-apoptotic effectors such as BAC and cause them to unfold and break apart membranes. So a little bit more on BAC. It resides on the outer mitochondrial membrane. It has its transmembrane domain that crosses this outer mitochondrial membrane, and under steady state, it's folded into this 9 alpha helic bundle, normally in association with the porin VDAC2. Upon a suitable apoptotic cue, we get upregulation or activation of a BH3 only protein such as BID. This can then go on and interact with BAC, causing it to dissociate from VDAC and unfold. These then unfolded BAC molecules can find one another, homodimerize, and then these homodimers form pores in this outer mitochondrial membrane that release cytochrome C. Once cytochrome C has been released to a sufficient level, that cell will eventually at some stage die, and typically triggers the caspase cascade. So we wanted to know, can we disrupt this process? Can we make design proteins that will bind to BAC and potentially prevent its binding to BH3 only proteins and stop this formation of pores? So this was all initiated by Stephanie Berger from David Baker's lab in the US. You may know her actually for this sort of poster child paper of her cytokine design proteins that target IL-23 receptors and IL-17, I believe, which are one of the first instances of design proteins being bioactive in disease. So what Stephanie has done is she took a previous protein called Bindi. This is a 3-helix bundle that has been designed to bind to a BCL2-like protein, BHRF1. She chose this protein because it had a high thermostability, and this allows it to be mutation tolerant. And she wanted to adapt this to bind to BAC, also BACs, but I won't talk about that in this story today. So this was initiated a while ago before these diffusion-based algorithms for protein design. So classical gradient descent methods were used, particularly in this case using Rosetta motif graph. What was done here was Stephanie took an experimental structure of BAC bound to a BID BH3 peptide, and using Rosetta motif graphs is able to sample all possible mutations of that BH3 peptide in silico to optimize the binding interface. You then take that peptide, graft it back into the Bindie scaffold. You then sample all possible mutations of the other regions of the other two helices around this graph that you have put into Bindie. And at the end of this process, get an optimized binder for BAC that in this case we call CDP02. So straight out of the in silico design process, this was quite efficient. The original CDP02 affinity for BAC was around 60 nanomolar, which is really quite impressive. However, in our case, we've got quite a challenge. You can't just bind BAC. You have to ensure that you don't bind other members of the BCL2 family. And this is because if you start binding to the BH3 binding groove in a pro-survival protein, you're going to compete with the binding for those pro-apoptotic BH3 only proteins, and that will actually promote apoptosis. And so one of the problems with this CDP02 was that it bound to the pro-survival protein MCL1 with very high affinity as well. So it turned out in our case that we needed a combination of both high potency in affinity, but we also needed high specificity for our target, in this case, BAC. And before I move on, just to show how impressive the 60 nanomolar was, the BID BH3 binding to BAC itself was typically around greater than 4 micromolar. So the standard, we've got quite a big improvement over the native binders here. So to overcome this affinity selectivity problem, Stephanie moved to deep mutational scanning. So this is an approach where you can perform site saturation mutagenesis on every position in this design CDP protein. And then you can rank these mutants for both affinity for BAC, but also selectivity for BAC over the other pro-survival proteins. This was done in 2 rounds. In the initial round, you take the CDP design, mutate every position to every other amino acid. You then score those, pick the ones that have the best combination of affinity and selectivity, create a new protein that we call anti-BAC1, and then through a second round, you then mutate every position in anti-BAC1 to get the final protein that Stephanie decided on, anti-BAC2, which I'll now go into in a bit more detail. So this anti-BAC2 is now what I'm going to continue talking on from now on. I won't confuse anything with anti-BAC1 or CDP02. This is the binder we're now discussing. It had a very high potency for BAC, 400 picomolar, and over than 300-fold selectivity over the other pro-survival proteins. So this is it here. Here's the CDP02, anti-BAC1, which is about 7 nanomolar, and then the 400 picomolar for anti-BAC. And you can see that it has this high selectivity. The closest binder was BCL2, but it's over 300-fold selective over this. So this is really where we wanted to be. But before we wanted to go on any further, we had to do a few things. First of all, we had to validate that the anti-BAC was binding at the site as predicted in the in silico models. At this stage, this was prior to Alphafold 2, so we didn't have a very effective way to accurately predict this binding. And then we also wanted to check that it had the affinity and was functioning as we expected. So this is where I kind of stepped into the project to validate the binding. So we turned to experimental technique, X-ray crystallography, and I was able to grow crystals of these heterodimers of anti-BAC bound to BAC. Now, when we solved the structure, it was actually quite surprising. It wasn't what we expected based on the in silico modeling. Um, the major feature change was that back had unfolded. Now, this doesn't normally occur. In order to do this, normally we have to treat back with a combination of either detergents or detergents and a BH3 only protein, whilst in this situation, it was happening with the anti-bac protein alone in the absence of any detergents, just in solution. Despite this, the anti-bac was binding where was predicted to in the back binding groove, and it was largely binding as predicted with the key residues that we expected to interacting where the design model had said. So this structure validated the computational design, but did bring into question what quite was going on here. Why had back unfolded and had formed this unlatched conformation that we normally only see when back is activated? What are the functional consequences of this? So in order to answer this question, we turn to a technique we use in our lab, specialized for looking at back breaking apart membranes. We use these liposomes. These are bilayers of lipids, mitochondrial outer membrane lipids, that we encapsulate a self-quenching fluorescent dye within. So when we activate back and cause it to form these dimers and higher order oligomers, this forms pores in the liposomes, allows the fluorescent dye to be released, and then because it's self-quenching, it's now released into solution and starts to fluoresce as it gets diluted. And we can use this as a tool to look at back breaking apart membranes. And this is what the data looks like when we add in anti-bac to these liposome assays. So here I'm representing the data as a percent release. So 100% is all the dye released into solution and 0 is no dye. And here we're titrating in across this x-axis various increasing concentrations of the anti-bac protein. The first thing that we noticed that was very surprising was normally in the absence of an activating stimulus, back would not break apart these liposomes. We designed anti-bac to be an inhibitor binding at the BH3 binding site in back, yet even at low concentrations we were seeing activation of back. However, when we hit a critical threshold, this activation of back started to drop off. In contrast, when we added in an activating stimulus, so in this case CBID, which is an activated form of BID that can normally promote back to release dye from these liposomes, we see that at low concentrations there's almost complete release of the fluorescent dye from the liposomes, because that back is causing that BID is causing back to release. However, when we hit that similar threshold around 30 to 60 nanomolar concentration of anti-bac, we started to see this inhibition like what we saw when there was no activating stimulus present. So despite designing the anti-bac to be an inhibitor by blocking the groove, it turned out that it was doing a combination of two things that were concentration dependent. At low concentrations, where we are below the concentration of bac that we have in the assay, it was functioning as an activator. But at high concentrations, when we have an excess of the anti-bac, it is now acting as an inhibitor and blocking those BH3 binding sites, preventing CBID doing its normal activity. So this was really quite surprising and was quite unexpected at the start of the design process. So that is where I want to leave the end of this first part of the story. And I've talked you through how we can use older style machine learning methods to computationally design back binders. The key in this process was to use site saturation mutagenesis or deep mutational scans to improve both the affinity and the selectivity of this anti-back binder for the back target. I've been able to show you that did manage to design and bind where we expected to. However, this actually had unexpected consequences in activity that we wouldn't have predicted prior to starting this project. And interestingly, there is this concentration dependence that probably has some important ramifications for designing back inhibitors in the future. Now, in the second part, I'm going to shift gears slightly and build on this by how we've been trying to use more deep learning approaches to redesign this anti-back binder to change its function in vitro. This was a project that was started between myself and Marjan, who was originally at WEHI but moved over to Monash to Gavin Knott's lab a couple of years ago. We were inspired by the Baker Lab's publications of hallucination and in painting when they first came out. And we got a small bit of money together from this Eve Marlab Award for blue sky research. So we came up with a really blue sky project of how we might be able to use these deep learning approaches to modulate our established back binder and change its function. The project was twofold. Can we establish these methods at WEHI? And then we came up with this pilot study of modifying anti-bac2, building in new functionality, which I'll go into in a bit more. Now, by the time we got the money together and got everything established, hallucination and in-painting got superseded by RFdiffusion that did a better job of both. And in fact, yes, this had come out by the time we probably managed to get hallucination installed in our HPC, which anyone who has worked with these programs over the last couple of years or so will know they can be a bit of a headache to get up and running on your HPC architecture. And so by the time we were starting to design, we were using RFdiffusion. Now, I know previous people have talked about this a lot in the previous webinars here, but I'll just briefly go over what you can do with RFdiffusion. It's using these denoising diffusion probabilistic models. Essentially, what you can do is use these to stepwise introduce noise into a protein structure. Because you know how you've introduced that noise, and what I mean by noise is moving tokens around in space. So whether that's an amino acid token or an atom token, and randomly distributing them into Gaussian noise. Because you know you've created that noise, how you've created that noise, you can denoise it to come back to your protein structure. And this can be used in multiple different ways. And in case that's most relevant to what we've done here, I'll talk about motif scaffolding, whereby you can set out a initial protein fold or architecture that you want to adopt by introducing more tokens or amino acids than are present in that initial description. You can then essentially denoise to a fold that will stabilize your architecture. And I have this sort of GIF kind of, which I feel is an loose approximation to what's going on here, whereby we have a picture of a horse. By going into random noise, we can take it out to a desired outcome. So switching from our white horse in a field into a robot horse, something that looks similar to what we started with, but we've guided towards a target. So the idea in this was we already have an established binder for BAC. This is our anti-BAC2, and it's pretty potent and selective. Can we modulate its binding to BAC? Can we create a new binder by installing new functionality? In this case, introducing a new domain for a small molecule that can block its ability to bind BAC. We did this in this. We had three objectives. One, we wanted to redesign the anti-BAC2 and retain its binding to BAC. Then we wanted to install this new domain that could bind a small molecule, in our case, rapamycin. And then the third, more sort of blue sky aim was, will this small molecule binding actually be able to modulate the binding of our anti-BAC to its target BAC? So the process that we came up with was using this FRB, FKB rapamycin binding as an established protein dimerization that is induced by the presence of rapamycin. We wanted to merge this into our anti-BAC2 structure. So I came up with this design model whereby I would loosely overlay the FRB and FKBP portions onto the anti-BAC2 in such a way that I felt could be integrated into the backbone of anti-BAC2, but also would potentially disrupt binding to BAC. From this, I came up with this initial design model that has portions missing. And the idea was, can we get RFdiffusion to use these diffusion probabilistic models to reconnect this design model into something that will actually fold together and hopefully have our desired outcomes of rapamycin binding, BAC binding, and modulation of binding to BAC in the presence of rapamycin. So in order to do this, again, this was quite a while ago when it was quite a challenge to install these things on HPC architectures. The standard Docker images weren't compatible with our HPC, so we had to install things differently. And this led to an approach that meant we ran the standard workflow, but we kind of didn't do it in the standard pipeline. We ran things individually. So we would run RFdiffusion, and then we would run MPNN in order to get our secondary structure and dock actual sequence that would adopt that fold. And then in the end would do validation with a full run of Alphafold 2 rather than using the standard Alphafold that's built into, not the non-standard Alphafold that's built into the standard RFdiffusion pipeline. So the first step was doing this RFdiffusion. We take our design model, I then run the inference pipeline. We say this is the command that was being run. So we tell which regions of the structure we want to keep, where we want to introduce new sequence, how long I roughly wanted those regions of sequence to be. And out of that, we would get a prediction of secondary structure. And we ran this, or Mariam ran this 3 times, asking for about 2,000 designs in each process. Then we would take this polyglycine model that gets spit out of RFdiffusion and then dock the sequence onto that using MPNN. And then we would take these sequences and run them through Alphafold 2. So this is an example. This is what comes out of RFdiffusion. You can see these large polyglycine regions of secondary structure that it's designed, and then MPNN docking on sequence onto this, that we then go and check, can adopt the predicted secondary structure using Alphafold 2. At the end of this whole process, we had requested 6,000 odd designs from RFdiffusion. due to the way we were running it, straight out of RFdiffusion, we could reject a lot very early on, just based on clashes at the rapamycin or back binding site, because these had not been included in RFdiffusion when we were making these designs. So we only ended up around 130 odd that we were actually running through Alphafold 2. And then at the Alphafold 2 stage, we ended up rejecting about another 90 based on the RMSD to the design model. And then the remaining, I was able to, remaining 40 or so, I was able to manually inspect and just look for things that made sense, things that didn't look right. Shortlist down to 11 based on that and a combination of sequence diversity and other properties that we might expect. The other thing we did was because this was quite a big challenge and we were trying to establish these techniques as WEHI, we wanted to make miniaturized versions of the anti-bac. This is a more simple task as you're just trying to create a binder and we already have an established binder. So here we made these shorter versions of anti-bac and from them selected from a 30, designing 30 through RFdiffusion in this pipeline, selected 4. Then the next process and probably the biggest challenge that we've had is then moving on to expression trials, principally in E. coli, then going on to batch culture, 1 liter scale and purifying, and then checking both binding to back rapamycin. We do this principally by SPR, and then checking the function in our liposome assay. So here is an alignment of the different sequences. Here we've got the dual binders. This to show we've got quite a lot of diversity between them. These are their Alphafold 2 predictions. And then these are the mini binders down here, and these are their Alphafold 2 predictions. So they all looked somewhat similar, but with, as you might expect, as we're going off a design model template, but they have built in large regions that are quite different. And it was unsure whether they would be able to bind back or the rapamycin target. So the first stage was expressions trials. This was really surprising for me. We were able to express all 15 of the proteins that we ordered DNA in small scale. So here we're talking about 6 mils just in Falcon tubes. We worked up this process where we would induce and then add in nickel beads and just do a nickel pull down to see if we got enrichment of our binders, and that's what we see here. These are our designed dual binders, which are quite large proteins, about 35 kilodaltons, and you can see various levels of expression purification for these. And then these are our mini binders down here, which have a size of about 10 kilodaltons. Again, we were able to express and crudely purify using nickel here. So from these, we wanted to simplify our process. So we took the designs that looked like they were expressing the best and took them to 1 liter expression. So from this, we selected 2 mini binders, and I believe it was 6 dual binders. This is where things started to get a bit more tricky, I guess. When we actually started to purify all these different proteins, we started to notice some weird characteristics to them. In size exclusion, we had various different oligomeric states for the different proteins. Some that were behaving as monomers, but we were getting poor yields. Some that we were seeing different levels of oligomerization. So in this case, something that probably relates to a trimer in solution. Some that were forming multiple different oligomeric states. Some that were dimers, monomers, and various different combinations of all of them. We often saw quite a lot of degradation as well. The mini binders, in contrast, were a lot simpler. We did tend to just see these as monomers, particularly this one here. Often we did see weird things happening as well, such as additional bands. We haven't gone on to characterize these higher order bands in detail, but they do appear to be some form of SDS resistant dimer of our design protein, as opposed to contaminating proteins from the E. coli expression. Despite this, we were able to successfully purify them. There's just caveats to their oligomeric status. However, there were no unicorns, I guess, in amongst this. We didn't get that one protein that expressed really well and didn't have some sort of degradation or oligomer that we weren't expecting. Despite this, we went on to try and characterize their binding. Here I'm showing SPR traces using predominantly single cycle kinetics. Here in SPR, we're looking at response changes upon binding of the design protein in solution to a BAC immobilized on the sensor surface. And as you can see, as you titrate in higher concentrations, these characteristic increases in response due to the dual binders, in this case, selectively binding to BAC. We got 3 that showed some quite nice binding with different kinetics. So, for example, some low affinity binders at around 10 micromolar that were fast on, fast off. One here that was a mid-range 1 micromolar binder with a slower on-off. And one here that was around a nanomolar binder that appeared to have some sort of conformational change going on in the kinetics, and definitely a slow off rate going on due to conformational changes. We also saw that other proteins here, they did show some indication of binding, but clearly the curves were not what you would expect, something sticky going on. So not quite sure what this is, but just to show that there can be complications with all these different designs. And then the mini binders, they again designed, they were quite good, not perfect fits by any means, but we got 2 different ranges of affinities, one at 4 micromolar and one at 120, which is not bad. So we then wanted to test the function of these proteins. So we returned to our liposome assay. So just to remind you, this is the anti-BAC2. These are our controls. If we titrate in from low to high concentration, what we would see with this dual function of anti-BAC2 is an initial activation. We see release from the liposome, but we hit a certain threshold, and then we start to see inhibition as we saturate the pool of BAC. With our dual binders, I've now reduced down to just 2 from our initial 11. We could see potent activation, but we didn't really see any inhibition, at least not in this concentration range that we were testing. But it was both potent activation for both of them. And the mini binders, we again saw a similar thing, potent activation. We didn't really see any evidence of inhibition. And this nicely correlated with the affinity. So this is the 4 micromolar binder, and this is the 120 nanomolar binder. We see that that is more potent than the weaker binder, which was quite reassuring. So of these 2 nicely behaved BAC binding proteins that are activating BAC, we wanted to see for the dual binders, do they bind the rapamycin that we had designed them to? And what was really quite reassuring was one of them at least does. So it has around a 250 nanomolar affinity for rapamycin, as shown here in these nice SPR curves. However, the other binder, although there's a hint here, the scale here is very small, so there's not really any binding going on here. Now, the final target milestone, I guess, for this project was to see if this would actually this rapamycin binding would actually modulate the binding of the design protein to the back. So we return to the liposome assay. We can see that here I'm showing an EC50 of the concentration of inhibitor against its release from the liposomes. And you can see whether you have rapamycin there or not in this protein that's not really binding to rapamycin, there's no difference. However, with the design protein where we do see rapamycin binding, there is potentially some difference. It's not striking. I'm not sold on this myself, but there is a fourfold difference in EC50 in this assay for binding, but this really needs more robustness to say that it's actually occurring. Nevertheless, we have been able to install a rapamycin binding site into a protein that can also bind to BAC. So that's all I've really got time to talk to today. In this second part, I've talked about how we have implemented a RFdiffusion workflow here at WEHI. This was actually established 2, maybe going on 3 years ago, and we've now sort of refined this pipeline now. Julia Iskander here in our research computing department has integrated it into Nextflow and Sakera. So we now have a nice pipeline for anyone here at WEHI to run RFdiffusion. Also in the next talk, you'll hear from Josh. He'll talk about how they've been doing a much more refined way of using RFdiffusion and a pipeline that they've established as well that you should definitely come along and listen to if you're interested in this. This project, we designed initially 6,000 jewel binders. We narrowed this down quite heavily to 130 after our MPNN, before MPNN. And then after Alphafold 2, we were able to further refine this down to around 40 designs before finally selecting around 11 or so for actual expression. We also created these mini binders. This was a lot more successful, where we were only initially asked for about 30 and selected 4 in the end. Expression trials surprisingly showed all 15 expressed to various levels. But it wasn't until we went on to this further characterization by size exclusion that we started to see problems and additional considerations that you should have when you're using these design proteins. From the 11 that we selected, 5 were confirmed to be, of the dual binders, were confirmed to be BAC binders. 4 were confirmed to activate BAC, and we managed to get at least 1 rapamycin binder. And there's some hint that this rapamycin binder can modulate binding. However, this is really like a pilot project that we put together to test out RFdiffusion. Methods have changed quite a bit now. You probably wouldn't use RFdiffusion in the same way to design a small molecule binder. So at this stage, I would like to finish my presentation. Before I go, just thank all the various people who've been involved in the project, particularly Stephanie and David, who were instrumental in the initial design of the anti-BAC. Here at WEHI, Peter Savitar, whose lab I'm within, various people who've worked on this, particularly Sila, Mariam, Yishua, and Cindy. They have been involved in obtaining crystals and doing liposome experiments and expressing proteins. A big shout out to our research computing team, particularly Julie and Miguel. They have spent so much time trying to get these software installed on our cluster. Marjan, who we got the initial award to start implementing these processes here at WEHI before she moved over to Monash, and NHMRC, the Australian Synchrotron And at that point, I would like to finish and open up to questions.
Thanks very much, Richard. That was a fascinating journey through your research. It was really interesting to hear how you're using these models not just to design proteins, but to refine them and adapt them and modulate their activity. If I could start with a question for me, and then we'll go to questions from the audience. If you do have a question, please write that into the Q&A box. You said right at the end that you probably wouldn't use RF diffusion in this way anymore. So if you were going to do this again, what would you change about your approach?
Well, so the methods have changed a little bit. MPNN, you now have an all-atom version of that. So you can use your binder, your small molecule binder, or the small molecule you want to build a scaffold around directly in the simulations. So that is the major thing that I would change. I would also probably go back and revisit how we would introduce that into the anti-bac scaffold based on these changes that will allow you to not rely on established structures to create your small molecule binding. I would probably also start using more refined versions of RF diffusion, such as Bindcraft and Protein DJ that Josh will talk about next week. Yeah, those are the sorts of things I would change, and maybe look into other ways to design binders, such as Alphaproteo, which might have a bit more success in getting the affinity you want for the particular site you want.
Sounds like there's lots of options and they're getting better and better all of the time.
Yeah.
Okay, so let's go to questions from the audience now. The first one says, great talk, Richard. And referring to the first part, can MD inform any of the potential back unfolding process during the design process?
Yeah, that's a really interesting question. We have not actually explored that, but potentially it could. I don't know how long those simulations would need to run for, but yeah, no, it's definitely an interesting question. One thing, it's something I always ask David and Stephanie about is if we could um, predict the unfolding based in Rosetta, and I have never fully understood their explanation, but it's a different challenge folding a protein to unfolding a protein. And essentially, Rosetta cannot do that. But maybe with MD, you would be able to um, address that. Yes.
Yes, I guess it's the opposite to what Rosetta is designed to do, makes it tricky.
Yeah.
Okay, next question is, could the multimer be successfully used to computationally screen out oligomer foaming designs, do you think?
Uh, Potentially, yes. So that was something we hadn't considered initially when we designed when I was making the initial designs, or when I say well, I hadn't really predicted, but it became somewhat obvious that that is something that could occur when you've got the way it was designed with this FRB, FKB building two components into one structure. One of the simpler ways that it would block binding is if, yeah, you have an FRB from one protein binding to the FKB of another protein, and that forming the dimerization to rapamycin. And rather than forming an internal conformational change that would block binding, you block binding just by oligomerizing it. Whether you could predict that with the multimer might be challenging, because you would predict that it's going to be rapamycin dependent. In the cases where you don't need rapamycin for this to occur, yeah, you could use multimer to filter them out. obviously you can now use Alphafold 3 to do this. I have folded these now with Alphafold 3 and rapamycin, and the 11 targets that we selected, they all, according to Alphafold 3, have the potential to bind rapamycin. We just don't necessarily see that
Mm.
in our in vitro experiments.
Which absolutely highlights the need to use these computational methods in parallel to your biological methods, because biology is complex and you never know what's going to happen sometimes.
Definitely. And I think that's sort of one of the take-home messages we've learned from this is that you can do so much in silico, but at the moment, the bottleneck is that translation between what happens in silico and what actually happens in your in vitro setting or in vivo setting, where it's just going to take time to build up that data, because it takes time to express and purify and fully characterize all these proteins at scale. Whether you can use things like Boltz-2 now to score your affinity for small molecules in this example, that's something we probably could have done as well. That may have given us a hint as to what would be a good binder, but yeah, that's something we now know in hindsight, I guess.
That's the only way you can learn in the end, really, trial and error. So a couple of more biologically focused questions now. Did you expect to design activators rather than inhibitors? And was there any evidence that they're unfolding back?
Yeah, so that was completely unexpected. I have presented it like we discovered that straight away. That is not the case. In fact, it was several years and battling to understand what was going on with this anti-bac protein. All the way along, we were told this was going to be an inhibitor, and we somewhat expected that based on where it was binding. But it wasn't until we started probing the biology a bit more that we really got a further understanding that this unfolding was happening. We've been able to show that by NMR that this happens in solution. And so it is definitely uh, so something that occurs and was unpredicted at the start of this. Um, this is something we are now looking to address in our sort of refinement of these binders, is how we can prevent the unfolding of BAC to make a true inhibitor, as opposed to something that has this dual functionality that's quite hard to translate into something that could be useful in the clinic, for example. Um, yes, I think that's all I have to say on that.
Great, thank you. And keep your questions coming into the Q&A box. So the next one we have is, did rapamycin affect the multimerization of the dual binders?
Yes, we haven't looked at that in detail. The ones we have looked at have been for rapamycin binding, we have chosen ones that are monomers, because it's just a bit easier to understand. But yeah, no, it would be interesting to look at that in more detail. We can say with those ones that it doesn't induce oligomerization. But for the other ones, whether it alters, where we're already seeing oligomerization in the absence of rapamycin, whether having rapamycin there changes that, would be interesting. Yeah.
Great, thank you, Richard. There's definitely lots of exciting different avenues that you can take here, and it'll be, I'm keen to see how this whole field evolves over time. We are going to have to leave it here for today. So if I could just take back the screen share, I have a couple of things to wrap us out. As was mentioned a couple of times, this is part of a series, and our next one is on the 7th of October. We'll be joined by Dr. Josh Hardy, who's going to be talking about protein DJ and how this was designed and how it's being applied in different situations. You can find out more about that webinar and all of our upcoming events on the Australian Biocommons website, or by following us on social media or subscribing to our newsletter. If you'd like to know more about the Australian Structural Biology Computing community, or even go along to some of their catch-ups and swap ideas about how to use these tools, you can find out more about them on their website as well. Last but not least, a huge thank you to Richard for joining us today and for sharing his research journey with us. And thank you to all of you for joining us as well. Finally, I'd like to acknowledge that the Australian Biocommons is enabled by NCRIS via Bioplatforms Australia funding. Thanks for joining us today, and we look forward to seeing you again soon. Until then, goodbye for now and enjoy the rest of your day.